Alrighty, hello everyone. Thanks for joining. We're going to get started here in a few minutes. Give a few minutes for people to roll in. Hi everyone. Thanks all for joining. We're going to get started here in about 1 minute after, just to give folks a minute to roll in. Thanks to those of you that just joined. We're going to get started here in about 1 minute. Alrighty. It is 10:01, so I'm going to get started just for the sake of time to keep on schedule. So hello everyone and welcome to the European Froglet Collaborative webinar series. We have an excellent speaker who has agreed to share his work with us today. Presenter Philip Weyl with the Center for Agricultural and Biosciences International, otherwise known as CABI, based in Switzerland, will be sharing his work regarding opportunities for the biological control of European frogbit in North America. My name is Nicole Angel. I'm joined by my colleague Taja Tucker-Silva with the Great Lakes Commission. On behalf of the EFB Collaborative, we want to thank you for participating in today's webinar. So before I turn the webinar over to our presenter, I wanted to provide some background on the collaborative. The European Frogbit Collaborative was convened in 2018 when the state of Michigan identified EFB as a priority and got funding from the U.S. Fish and Wildlife Service's Great Lakes Restoration Initiative grant with Central Michigan University. Once the initial supporting structure was established, an adaptive management framework was developed, and CMU was established as a technical advisory group. Finally, stakeholders in Michigan were engaged and encouraged to participate, and initially this work was Michigan-centric, but as EFB became a larger issue, the need for this type of framework grew. And that's when the Great Lakes Commission was identified to provide a neutral backbone for this framework in 2021, and was subsequently awarded funding through GLRI in 2022 to continue to expand the EFB collaborative framework that was put in place by Michigan. So here's a visual depiction of the governance structure for the EFB collaborative. There are currently 64 members as part of the collaborative coming from various organizations across the Great Lakes Basin. The steering committee provides strategic guidance and directs the collaborative's work, as well as speaks on behalf of the collaborative and makes decisions on what to work on. There are 3 work groups comprised of subject matter experts that focus on specific aspects of invasive European frogbit, including prioritization, management assessment, and education and outreach. The collaborative membership at large is represented by the white box, and they give input on all the products that the collaborative develops. And then, as prior mentioned, the GLC provides neutral backbone support to the collaborative, including facilitating meetings and keeping everyone on track to achieve goals and objectives. So the European Frogbit Collaborative established its regional strategy, which serves to guide the work of the collaborative from 2024 to 2026. The strategy is founded upon 3 core focus areas of regional coordination and collaboration, best management planning, and research and information needs. And this strategy really helps shape what the work groups focus on. So I wanted to go back to this structure diagram and quickly highlight some of the recent accomplishments of each of the work groups, starting with the prioritization work group. So the prioritization work group was charged with reviewing information on where EFB is impacting Great Lakes resources, identifying criteria for evaluating sites based on this information, and then developing a system for prioritizing sites for potential management activities. This work group is currently working on a demo version of a new tool, which you can see pictured on the screen. And this tool is going to help managers prioritize where to both monitor for and manage EFB. Moving on to the management assessment work group. This work group, the goals of this work group are really to develop a tool that managers can use to easily track their EFB monitoring and management actions, and then use that data to help determine what best management practices for EFB are. So this work group has recently developed a new field maps application to accomplish these goals. It's called FrogBat, the before and after treatment tracker. And the first summer of field trials of this app are complete, and an SOP will be released soon. Next, moving on to the education and outreach work group. This work group was charged with informing, developing, and publicizing future EFB educational materials at a local and regional scale. Recently, this group has organized and hosted successful EFB-related conference sessions and a social at the Upper Midwest Invasive Species Conference, and they're working to develop fact sheets such as the lookalikes document that you see on the screen, as well as a fact sheet explaining what someone should do if they think they have spotted EFB. And then finally, I wanted to mention the delimitation work group, which wasn't pictured before in the overall structure of the collaborative. Because this work group has completed its objectives and has since been phased out. But it's important to mention that this work group developed a delimitation application where managers can report where they have seen EFB as well as where they have monitored for it. And then I just wanted to end my introduction by saying that there's lots of ways to engage with the collaborative community if you're interested. We have a website that hosts all the archived literature and resources developed, a quarterly newsletter with the latest and greatest EFB information, as well as an amazing collaborative mascot named Froggy, which you can see shown in the middle of the screen. If you'd like to learn more about the collaborative or are interested in staying in touch with our communication networks, we encourage you to visit our websites and join our listservs. And then, while we always strive to minimize technological issues during these webinars, we do apologize for any that may arise. Before we get to our presentation, I'd like to cover just a few logistical items. So we've automatically muted all attendees to reduce interruptions, and attendees will remain muted with webcams disabled throughout the webinar. However, we do encourage and will accept written questions from attendees during the webinar. If you have a question, please use the Q&A function to do so. Questions may be submitted at any time during the webinar and will be addressed after the presentation has been given. If you're experiencing any tech difficulties, please use the chat box for that or email Taja, whose email is shown on the screen. Alrighty, and it is now my great pleasure to introduce our speaker, Philip Weyl. After completing his PhD with the Center of Biological Control at Rhodes University, South Carolina in 2015, working on the origins of a submerged macrophyte, Myraphyllum spicatum. In Southern Africa, Philip worked on a postdoc position with the CBC. Philip started with CABI in 2016 as a research scientist in the weed biological control section, and in 2023, he started leading the team. This role involves managing approximately 20 projects, mainly for the United States and Canada, but also for Australia and South Africa. Philip remains active in student supervision, with master's and PhD students at the University of Neuchâtel, Switzerland, CBC, Rhodes University in South Africa, and the University of Idaho in the United States, where he is also an affiliated professor since 2020. And with that, I am going to end my slides. And Philip, you can feel free to start sharing yours and take it away. Thank you, Philip.
Oh, thanks so much. Now, let's see if this works, eh? Okay, I'm assuming that's working.
Yep, looks great.
Okay, thanks so much for the introduction, and it's really nice to see so many people joining the webinar. Very much appreciated, and I'd just like to thank the European Frogbat Collaborative for the opportunity to speak today. I'd also like to just mention that this presentation is, this project is funded by the Army Corps of Engineers, and our contact there is Dr. Ian Knight, which I'm sure many of you have met or have heard about before. So again, just to mention that this is a presentation put together by the research team that's involved in the biocontrol project at CABI. So that includes Patrick and Laureline as well. So I just wanted to give you a bit of a background to CABI and where we sit. So we sit in the northwestern part of Switzerland in the canton of Jura on the French border. This is an aerial image of the institute. So we've got our labs and offices up here on the top left, and then we've got various greenhouses, garden beds, and this huge area of land for various open field tests, cage experiments, and so on. I would also like to mention that this photo is a little bit outdated. It's missing all of the pools in the bottom left here and up here, which we've now established for both the flowering rush and European frogbit research. So the weeds team at CABI in Switzerland is led by myself, and then we have 4 scientists based at the Swiss Center. We have another in Serbia, Ivo Tosevsky, as well as technical staff, and then Meng Sun, who's a visiting researcher, and Chiara, who's the last of our summer interns for this year. Okay, so our work at CABI in Switzerland largely revolves around weed biological control, or at least the team that I lead, where we're involved in foreign exploration for potential natural enemies. We also do risk assessments, so host range testing, trying to determine the risk to native plant species. And coupled with that, we do a lot of work on improving protocols for testing and risk assessments of biocontrol agents. We're in the unique position that often the agents we work on are native, so we can do realistic impact assessments, and then ultimately we help developing the petitions for field release to get those biocontrol agents from the native range released into North America to start controlling the weeds. Okay, so it's quite a long list of projects. I mainly put this up, so if anybody is interested in some of the other projects we work with, please feel free to reach out. I'll gladly give you any additional information. But I would just like to highlight the new targets, which includes European frogbit, water soldier, and then Echium vulgare. The first 2 I'll be speaking to in a bit more detail in this presentation. Okay, so CABI in Switzerland is not, we're not isolated. So we are part of a larger intergovernmental organization, which is not for profit. CABI was established by United Nations treaty level agreement. We are not owned by a single country or entity, but rather our member countries, each of which have an equal role in the organization's policies, strategic direction, as well as governance. And then we, as an organization, address issues of global concern, the group that I'm involved in, the invasive species, but also in food security and food safety, and that's largely through our research and international development cooperation. So that's the research side of things, but we also have a major publishing, or we are a major publisher of scientific information, and that includes books, ebooks, online resources, the CABI Compendia, for example, something that if you don't know about, you should go and check out, the Invasive Species Compendium. We've got a lot of information on invasive species. So this is a list of our member countries currently. And what I quite like about this slide is it shows the whole range of countries that are part of our membership. And those include developing countries such as Bangladesh, Malawi, Zimbabwe, all the way through to developed first world countries such as Switzerland, Australia, and Canada. So it's quite a cool wide range of countries that form our membership. In addition to that, we have a global reach. So we sit in the Swiss Center, but we also have roughly 450 plus staff across 26 locations, 11 of which we have actual CABI centers. And then the UK team is also involved in weed biological control. Okay, so enough about CABI, and let's get onto the fun stuff. So I'm sure majority of everybody on this webinar is aware of European frogbit, but in case there's one or two of you that are not, I thought I'd just give some basic information. So European frogbit, or Hydrocharis morsus is a floating macrophyte. It can be rooted, especially when it grows in shallow water. And it has this positive association with emergent species, and that's largely because individuals get trapped, and then as they produce ramets, that population grows. So it is dioecious. Now, seed production in the literature is quite conflicting, whether they produce seeds or not. But what we do know is that when they do produce seeds, in many cases, over 90% of those seeds remain viable. So that's a massive propagal pressure if those populations are producing, if they're producing fruits and seeds. The overwintering stage is turions, and this is a photograph up here in the top right-hand corner of what the turions look like. They can produce up to 1,500 turions per square meter, which is an absolute massive number. And if you couple that with a single turion being able to grow to cover a square meter in one growing season, you can just imagine the exponential growth in the invaded range. So European frogbit prefers organic substrates and typically meso to eutrophic water conditions. This is just a photograph down here of what these infestations can look like in the invaded range. Okay, so just some important dates for European frogbit. So it was first introduced, or the first recorded introduction was in 1932 as an ornamental species into Ontario, Canada. And in the literature, it's recorded that it came from the Botanical Gardens in Zurich, Switzerland, which puts us in a unique position that we are very, very close to the potential point source introduction. It was first observed outside of cultivation in 1939, which is not too surprising. It's quite an invasive species that's able to escape cultivation quite quickly. And then in 1974, it was first observed in the US near the St. Lawrence River. So it is part of the Hydrocharitaceae family, which does include other serious invaders such as Hydrellia species, and Egeria, but also several native species such as Aloides and Vallisneria. So this is the distribution within North America, largely around the Great Lakes system. And then if we have a look at the distribution within the native range, it's got a massive distribution all the way from Western Europe, in the United Kingdom, through onto the continent of Europe, up to Finland and Norway, and then all the way across the top of Kazakhstan and into Russia. And I'm sure it's probably further south. It's just these are not recorded in the on any databases available to us. Okay, so why do we want to manage frogbits? And I'm sure many of you are more well-versed than myself in this. And in the native range, it's really not a problem. Whereas in the invaded range in the US and in Canada, you're needing to go out into these lakes and water bodies in canoes and absolutely filling them up with European frogbits. So management to date is largely focused on harvesting and shading, but there has also been some chemical control, and we can see that evolution of the different chemicals being used from the 1960s into the 2000s. So what we know about biocontrol so far is that native North American herbivores, which would largely be generalists, are not sufficiently damaging to control European frogbit. So we do need to look at other options. Grass carp, I'm not a fan of grass carp personally, but there have been some mixed results using grass carp to control European frogbit. Sometimes it works, sometimes it doesn't. Not very consistent. And currently, there are no classical biocontrol agents available for use. However, this is what we've kind of started this year, is to get out into the native range to search for potential biocontrol agents and to develop those. And then we all know why we want to control European frogbit, and that's largely because it has these negative impacts on vegetation, on dissolved oxygen concentration. They impede our boat traffic and various recreational activities. So with funding from the Army Corps and Dr. Ian Knight, he's put together this kind of work unit approach where there's essentially three main prongs. So we've got the foreign exploration, which is largely run by CABI in the native range, and that's for natural enemies. But what's also happening in the background, and I know that many folks at the European Frogbit Collaborative have been involved in collecting samples for genetic characterization across the native and invaded range, just so that we can try and identify where these plants originate from, which will hopefully guide us in our foreign exploration activities. And then Dr. Ryan Versal is also looking at herbicide screening to update that management guidance in North America. But what I'll be talking to you is about this first point here, is the foreign exploration for natural enemies. Now, I just wanted to get everybody onto the same page as to what classical weed biocontrol actually is, in case there's any doubt. So it's the deliberate use of specific natural enemies to control an exotic weed. And if I can just put this into context, so we've got the same plant species in the native range and in the introduced range. So in the native range, typically the plants they might be smaller. They certainly won't be as densely populated, and they're generally not a nuisance species. Where in the invasive range, or in the introduced range, the plant is considered invasive. They may be bigger individuals, or certainly more dense kind of monocultures, and they are considered a nuisance species. Now, if we think about classical biocontrol, typically in both ranges, you may have a suite of generalist natural enemies, and these are not specific. They might be crambid moths in our case, or various grasshoppers and things like that. They just kind of feed on just about anything green. And you may have representatives of both in the introduced and in the native range. Typically, these are not damaging enough to have any kind of population or individual level impact. But what is different is that in the native range, we often have a suite of specialist natural enemies co-evolved and adapted with our target plant. And they are capable of exerting enough pressure on the population or on the individual to have a kind of effect or controlling effect. And what we'd like to do with classical biocontrol is to reintroduce those specialist natural enemies in the introduced range to have that population level effect and bring those populations down. So the goals of weed biocontrol are to permanently reduce the weed density below an ecological or economic kind of damage threshold. Now, what's important to understand is that weed biocontrol will never eradicate a weed population. And then coupled with that, we'd obviously like to restore these native species communities and bring back that native biodiversity. So if we have a look at this graph here, so we've got population density of the weed here over time. And then at this population density, we consider the damage threshold. And this might be having an effect on aquatic biodiversity or having an effect on irrigation. Now, typically our weed density is above that, and it's considered a nuisance species, and that's why we're all here today to figure out how to bring this below that damage threshold. So once all of the testing is done and an agent is considered safe for release, we would release that agent. And what we often see is the biocontrol agent density may build up relatively quickly, and this is due to the fact that there's no limiting resource. The weed density is so great that the biocontrol agent density can increase rapidly. And again, this is the ideal scenario. And then what we see is that the agent is able to bring that weed population density below the damage threshold. And then we've got these sort of natural oscillations in the population, and that's essentially just due to a kind of predator-prey life cycle. You've got good years versus bad years of both the agent and the target weed. So in a nutshell, that's kind of weed biocontrol and what we would like to try and achieve. And then, of course, bringing back that desirable vegetation and biodiversity. Okay, so this is thanks to Dr. Nate Harms for this development pipeline. I quite like this image. It quite nicely breaks down a kind of biocontrol project development pipeline. And where we're at the moment with the European Frogbit project is really in the native range survey. This is labeled quarantine in our case. Because we're working in the native range, this will just be essentially lab work. So the native range surveys may include things like genetic surveys, exploration for herbivores, field observations of impact. We may also include surveys within the introduced range, which Dr. Ian Knight has been involved in. And then sort of the quarantine or lab work, we'd be looking at sort of safety studies, host range testing, understanding basic biology, and then hopefully in due time submitting those documents for release. Okay, so in terms of the steps, just to reiterate that, and largely CABI is involved in the first part of the testing. Once it goes to mass rearing and release, that would be largely handed over to North America, for example. But we're involved in things like weed identification, weed life history, which include things like the biology, origin, and distribution, survey for natural enemies. Once we've got a bunch of natural enemies, we then select potential agents and study those as well as their biology as well as risk assessment. Now, when we were looking at the weed life history, what we realized is that, so this map down here shows the distribution of European frogbit. And what we realized is that water soldier or Stratiotes aloides really the distributions overlapped, and many of the site-specific, there was also many sites that specifically overlapped as well. So we were able to convince Army Corps to allow us to survey water soldier as well as Hydrellia or European frogbit. So just to give you a quick introduction to water soldier. So it was first recorded in Canada in 2008. It's not, as far as I'm aware, it's not yet in the US, but it is making its way down towards Lake Ontario. It forms these dense floating mats. It is an awful plant. It has these serrated, hooked edges. It is a nightmare to work with, and most management at the moment is limited to physical removal. I do feel for those people. It is not a fun plant to work with. There is a little bit of herbicide usage, but not much. In addition to that, it is also allelopathic, so it does alter native plant communities, and it's likely to have quite a large impact. And then in the native range, what's quite interesting is that it's rather uncommon. It's on many red lists and potentially a endangered species, but it does co-occur with European frogbit. Okay, so to get onto the biocontrol, I would also like to mention that we've only started this project this. We're very much in the early phases, but we've made some really nice progress. So from the literature survey on both species, we have 15 herbivores. Unfortunately, most of those are considered polyphagous, so not necessarily useful for biocontrol agents. However, we do have two groups of species which we prioritized, which are often monophagous, the Hydrellia flies and the Bagous weevil. So this is a Bagous weevil here, and I'll talk to you a bit more in more detail with you on that in a moment. Now, many of the species are recorded both on European frogbit and water soldier. However, if we dig down into the literature, these are quite often doubtful records. So the species are often not reared off those plant species, but may have just been recorded in a site associated with European frogbit and water soldier. And if you remember what I said a little bit earlier, that In many cases, these plant species co-occur at the same site. So often surveys would have collected a bunch of insect species and then associated those with plants recorded at the site. We have very little information on individual species actually reared from those plant species. Okay, so our field surveys this year, so we were able to visit 48 sites of European frogbit and 12 sites for water soldier. This is the map here on the right-hand side is our distribution of sites at the moment. So what we did was we really focused on northern Switzerland, around the Zurich Botanical Gardens, and relatively close to home. And then decided to go further north. So we had sites in France, in the Netherlands, in Germany, on the Polish border, and then we were able to also survey in Hungary, Serbia, and in Greece. So now at each of these sites, we're collecting genetic samples, and that's to try and determine the region of origin of the North American invasions, and we're sending those over to Ryan Thum. And then at some of these sites, we're also collecting demographic data, and that is to compare with the North American demographics to try and identify which potential life history or parts of the plants that might be the most useful to search for biocontrol agents. For example, if seed production is relatively minor in North America, if we were to find a seed attacking agent, that may not be particularly useful. And then, of course, at all of these sites, our main goal is to survey for herbivores. Okay, so where are we? Like I said, we've prioritized the Hydrellia species. Now, it's incredible. This is by far the most widespread herbivore on both weed species. We've recorded them from majority, if not all, sites that we visited. Now, there's 6 species recorded in the literature, but according to the taxonomist in Germany that we communicate quite regularly with, there are only 2 monophagous species associated with European frogbit and water soldier. All other records are most likely misidentifications, and the species that he says are monophagous are Hydrellia albifrons on European frogbit and Hydrellia torsata on water soldier. At each site, we are collecting genetic information, as well as sending adult flies that we rear from each plant species off for identification, to try and prove that these species are in fact monophagous and only 2 of them, not 6. Now, our main target for this project is European frogbit. Like I said, with the water soldier, we are just surveying to put us in a good position to start a biocontrol program, but it's certainly not a priority in terms of additional testing. So with the Hydrellia that we've collected from European frogbit, we've been able to rear 2 generations in the lab, which is far more than I had anticipated starting out with this project this year. We've learned a lot about their biology, of which much was unknown. So we know that the eggs are laid on the leaves, and the larvae primarily mine in the leaves. And if you have a look at the top left-hand corner, this is a leaf that's been mined by a larvae. And in fact, you can actually see the larvae inside the mine over here. So a single larvae will start a mine, and they'll typically move throughout most of the leaf, breaking up that surface, also reducing the ability of the leaf to photosynthesize. When the larvae is done mining, pupation takes place typically in the petioles when petioles are available. If for whatever reason the petioles are either too thin or not in a good enough, if they start going brittle or rotting, the larvae will quite happily pupate on leaves or on any other structure that it can find. Now in the lab, development time takes typically about 3 weeks, but in reality, out in nature, we would expect that to be sort of closer to the 5 or 6 week mark. But that does mean that the species will have multiple generations over the summer period, and those populations will hopefully be able to build up fairly quickly and have an impact on kind of a population level impact on the mats. We're still unsure what the overwintering stage is. We're pretty convinced at the moment that it's pupae, so we went through a couple of our rearing containers, and we found a bunch of pupae, but this needs to be just sort of floating in the water. This needs to be confirmed next spring when those adults emerge from there. But at this stage, we still need to identify that overwintering stage. Okay, so moving on to the Bagous species that we also prioritized. So we found adult weevils at 2 sites, one on European frogbit in Hungary, which is potentially Bagous puncticollis, and one on water soldier in Germany, which is potentially Bagous binodulus, which is great because puncticollis has been recorded on both European frogbit and water soldier, but so far we've only found it on European frogbit. These have been sent for identification, and we'll hopefully confirm those IDs soon. So while we were in Germany, we also found 11 Bagous eggs in petioles of European frogbit, which got us extremely excited. We didn't find any adults at the site. We were able to hatch those larvae. They began mining in the petioles of European frogbit, but none of these developed past the second instar. Now, what this probably means is that this is a different Bagous species. It's virtually impossible to identify them from first instar larvae, but it's probably a different Bagous species associated with another plant as a preferred host. Europe has roughly 60 Bagous species, 60 native Bagous species, each one associated with a different aquatic plant species. So it's quite possible that this could be one from an elisma or another aquatic plant at the site. And those eggs were laid, I have no idea why, in error or that's just where those species lay their eggs and then the larvae move on. We've got some for genetic analysis, so we will hopefully be able to resolve that soon. Okay, so our plans for 2025 and going forward is to start developing a test plant list of the Hydrellia in North America. And if there's anybody on this call that is in a position to help us with that, either a botanist or somebody who may have access to Hydrellia plants that could send some over, I'd be very keen to talk to you more about that. Because we've only found Bagous weevils at 2 sites and only 1 site, we found Bagous puncticollis, which is the Hydrocharis or the European frogbit Bagous. We'd like to do a more targeted search for that, so return back to Hungary to try and collect more individuals, but also go to Sweden as well as other countries with recent records. We did not record any pathogens this year or any interesting pathogens this year on European frogbit, so we'd like to spend a bit more time doing that. And then, because we have a rearing of the fly in-house, we would like to start some preliminary host specificity testing with the Hydrellia species on European frogbit to try and determine whether these species are in fact monophagous to European frogbit or if they feed on other species. In addition to that, we would like to start looking at trialing kind of impact experiments with the Hydrellia species. There's often doubt whether Hydrellia species are in fact damaging enough to have a kind of population level effect. So we'd like to prove that before we invest too much time and effort into the species. But looking at impacts in the field, I'm pretty convinced that these Hydrellia species are impactful, and given that they can have multiple generations per year, I would hope that those population levels would be able to increase quite rapidly during the summer period. I'd like to thank you for your attention. I'd also just like to thank our funders, the United States Army Corps of Engineers, and Ian and Mike, who were able to come out. So this is Ian Knight over here, and Mike Greer, who were able to come out over the summer to spend some time with us in the field. Yeah, thank you very much.
Great. Thank you, Philip. I am just going to take over.
If I could just mention, so there's my contact email. If anybody has any questions or would like to reach out or would like to join the project, please feel free to drop me an email.
Wonderful. I'm just going to take over screen sharing here.
Yeah, absolutely.
And again, just say thank you. That was amazing. And that concludes our presentation for today. And we're going to move on to answering any questions you might have. As a reminder, please use the Q&A function to submit your questions or leave a comment. If the Q&A is not working for you, do feel free to use the chat. So let's see if we have any questions. Looks like we do have one, not EFB related, but water soldier related. Can you speak to the genetic samples collected for water soldier and potential lineage? Are you interested in additional samples from new site locations? And just for reference, it's from Francine McDonald with OMNRF in Ontario.
So yes, to answer the second part of your question, yes, absolutely, we're interested in additional samples from new locations. In terms of speaking about the potential lineage, so all we've done at the moment is collected those genetic samples. We actually haven't got any results on the analysis of those yet, but I'll gladly keep you in the loop as we do get those in. I think would you be able to send me an email and we can talk in more detail about where to send samples, how to collect them, and that kind of thing? That would be great.
Yeah, and maybe Philip, if you want to put your email in the chat, I sort of took away screen sharing from you relatively quickly. But yeah, happy to make that connection. And then I had a quick question. So I know this is the first year of the project, but once you see this project coming to fruition and, you maybe potentially square down on which is the best control agent, would you then make recommendations to organizations in the US and Canada, or like just maybe to help with the approval process or that sort of thing? I'm just curious how that works logistically.
Yeah, so this is where it gets a little bit not complicated, but it gets, so we do need collaborators in North America. So being an international organization, we cannot submit any petitions for release. That has to come from internally. So typically we would have a consortium, and that's individuals that are interested in the project, who would help us in writing that petition and then ultimately submitting that to tag to the either to tag in the US or to the Biocontrol Review Committee in Canada. And then the permission for release or the permits for release would then go to that individual, to that group of individuals who were part of the project, and then they would take that forward for releasing as biocontrol agents. We are very much in the early days at the moment. We're working very closely with Dr. Ian Knight with the Army Corps. But we would be very interested in getting other people involved, potentially other funders as well, to speed the project along. Essentially, unfortunately, it always comes down to funding. But the more funding we have, the faster these projects can go, because we can invest more time and efforts. Yeah.
Awesome, thanks for that. We have another question. Can you please share more of your thoughts on the relationship between EFB and water soldier? Is there a symbiotic relationship? Does water soldier provide wave attenuation, other things?
That's a very good question, and I don't have a very clear answer for that. So we often find, the two species in the native range at very similar sites. Often associated with each other, but if there's a kind of symbiotic relationship, I cannot say with any certainty. I would have a feeling that it's probably just due to it being nice habitat, and that both species are able to coexist at those sites. We do often find European frogbit at sites without water soldier and vice versa. But quite often they do coexist. But I think it's more habitat related than a kind of symbiotic relationship. But that's very much a guess. I don't know for sure.
Great. Just checking the Q&A and chat here. I'm not seeing any more at the moment. And I'll just fill in some time here. Your funding is mainly through U.S. Army Corps, correct? Is there any other funding organizations? Just thinking of how, if someone wanted to support, like what organizations your funding is through.
Yeah, so currently only the Army Corps. So we are okay. So maybe I should give you a bit of the funding strategy that we like to have at CABI is to kind of have a consortium approach, and that really allows us to have longevity of projects. It also means that often any amount of funding is not, there's never too little funding, if that makes sense. So quite often biocontrol projects, I don't want to say expensive, because if you compare them to like developing a herbicide, that would be, biocontrol would be a drop in the ocean. But it does cost money to develop biocontrol agents. And often, funders or donors might think, oh, I've only got sort of 5,000 to contribute. That's not really enough to get anything off the ground. But because we have a kind of consortium approach where we have multiple donors funding a single project, any amount is not too small, if that makes sense. What it also means is that often our projects, because they take so long, if you think of that kind of pipeline that I showed, it's anywhere 10 years plus. I mean, some of the projects we've been working on, like Phragmites, which I'm sure you're all aware of as well, has been going since the 90s. And we're just starting to, well, not just, but we now finally releasing agents some 25 years down the line. But what it means is that if funders only fund 1 or 2 years, or they don't have, so what we need is continuous funding for upwards of 10 years, and having a consortium approach means that funders can kind of come and go, but we manage to keep that project ongoing. So multiple donors for a single project are incredible. I can't stress how important that is for the longevity of projects.
Good to know. Let me just check the chat, and we do have one more that came in. You mentioned interest in receiving samples of North American Hydrocariaceae family species for testing. Is there any guidance for sending specimens and I'm thinking it's to CABI. Should we follow up with you directly?
Yes, please follow up with me directly. It's relatively easy, but what we do need is a phytos. So firstly, I should mention it depends which species. If it's not a species that's blacklisted in Switzerland, then we're good to go. But what we would need is a phytosanitary certificate, and that depends. That depends on different states and different contexts. But if we can get that phytosanitary certificate and it's not a regulated species in Switzerland, we can essentially put those in a FedEx package and send them over. If it is a regulated species, we can get essentially a letter of authority, which allows us to bring it into our quarantine facility. So we can do some testing. It just means that we'd have to test those species within our quarantine facility and not in the lab or outdoors. But yeah, there is some guidance, and I'd gladly chat to you in more detail about that, depending on which species you have and how you may be able to help.
Awesome, thanks for that. Not seeing any more questions in the Q&A or chat. So I'm just going to, again say, oh, I thought I saw one roll in, but I don't think so. I just want to again say thank you so much for your presentation. And for everyone who joined today's webinar, the webinar is recorded and will be posted on the European Frogbite Collaborative website in the next few days. So if you have any additional follow-up questions or comments, feel free to email us at the Great Lakes Commission or Philip directly using his email in the chat. And with that, I'm going to end the webinar and thank you all for attending.
Nicole, just one thing while everybody is still on. So I might use this as a bit of a advertising. So with NAISMA, there's a biocontrol summit happening tomorrow on aquatic weed biocontrol, which might be of interest to many of you. I'm going to put the link in the chat for people to register if you haven't done so already. It's usually quite a nice event. It's quite long, but we'll be covering biocontrol of aquatic weeds from kind of east to west, north to south in North America and Canada. So it's a nice update.
Wonderful. Yes, I know I am registered for the Biocontrol Summit, and so are some others at the GLC. So we're excited for that. Thanks for the plug there.
Great.
But with that, I will close out the webinar if there's nothing else. And everyone have a wonderful Wednesday.
Thank you.